Indian Institute of Chemical Biology was established in 1935 by a group of patriotic clinician scientists. It was integrated into CSIR in 1956 and renamed as the Indian Institute of Chemical Biology in 1982. The second campus of CSIR-IICB was inaugurated by the Honorable Minister Dr. Harshvardhan at Salt Lake City, Kolkata in 2015.
Indian Institute of Chemical Biology is a premier institute of CSIR. CSIR-IICB, since its inception, conducting multidisciplinary efforts to conduct research on infectious diseases, specifically leishmaniasis, along with the development of technologies for the diagnosis, immunophylaxis and chemotherapy of diseases. From last 15 years, our scientists at IICB had been working in the area of new degenerative diseases, cancer therapeutics, antiviral therapy, cancer cell therapy and development of natural products and novel small molecules against cancer and multi-drug resistant bacteria. IICB team of scientists are trying to take small molecules, biologicals from bench to bedside.
Recently, we have developed Alzheimer's disease detection system which uses brain MRI image processing and deep learning system. This system is capable of detecting Alzheimer's disease and especially the milder and earlier version of it. We have also developed a web platform named CCAD which uses brain MRI images and corpus callosum atrophy based differentiation between AD patient and healthy ones. This system is also available online. In addition, we are also looking at brain stroke and trying to identify brain hemorrhagic stroke using brain CT images, image processing and deep learning. Our primary objective is to identify the strokes and then convert this stroke region into 3-dimensional reconstruction and further connect this stroke, the volume and spread of the stroke to the outcome of the stroke diseases. It will be helpful in clinical decision making if we could actually successfully implement this. Thank you.
One of the main focus of CSIR-IICB is cortisol therapy. The cortisol therapy has many advantages compared to traditional cancer immunotherapy because it has very few to no side effect. Very importantly, it is like a living drug. Even though cortisol therapy first clinical trial started on 2012 in USA, but India could not do and it is lagging behind for many reasons. One of the two major reasons for this lagging behind is poor infrastructure and insufficient clinical training by clinician in the GMP facility. Understanding these two big challenges, and we made a team. That team consists of CAR-T designer, in vitro validator, in vivo validator, finally a clinical team. So, we made a team like IICB, IGIB and Jamia Millia and Apollo Clinical Hospital. This team had made a various strategy to take care of both liquid cancer and solid cancer. We use a multiple strategy and this strategy will be explained by my colleague Dr. Amit Srivastava.
So, the major disadvantages of currently approved CAR-T cell therapy are it's individual tailored. That means the one CAR-T product can be used for one patient. And another thing is, it's a very expensive, and it produces severe toxicity. So in our approach, that we want to utilize the off-the-shelf CAR T cell therapy approach, where we are going to use T cell iPSCs as a source of T cell. So we can get the unlimited supply of the T cell and can reduce the CAR T cell therapy cost. And apart from this, we have signed a MOU with one startup, Cellogen Private Limited, Delhi, and where we are developing bi-specific CAR T cell. So the one CAR T will target 2 antigens on the cancer cells. So it will give more therapeutic efficacy as compared to the single specific CAR T cell. And moreover, we have also formulating new projects with NIBMG Kalyani and AIIMS Kalyani for the development of CAR T cell therapy specific for solid tumors. So this is our efforts in the direction of CAR T cell therapy research.
I am pleased to share exciting developments in the ongoing scientific pursuits in IICB, funded by Swasthaniketan Integrated Healthcare and Research Foundation, a Bangalore-based startup company. The project is aimed at identifying an Indian variety of a medicinal plant for providing palliative care and treatment to cancer patients, akin to its European counterpart. India, with its rich biodiversity, presents an ideal environment for such exploration. Our team of natural product chemists has embarked on an extensive study to characterize the chemical constituents of European variety and find similar varieties of the plant native to India. The team at Swasthaniketan is meticulously collecting specimens from different regions of the country. The collected plant samples will be subjected to comprehensive chemical analysis at CSIR-IICB by our team.
Early results from our chemical investigation indicated the presence of certain phytomolecules known to have anti-cancer properties from the European varieties. Discovering an Indian variety having similar chemical profiling with anti-cancer potential could open a new avenue for the treatment of cancer patients and contribute to the growing field of natural medicine in the country. The ongoing pursuit of identifying an Indian variety with anti-cancer properties is a testament to the power of interdisciplinary collaboration between traditional knowledge and the modern science of the country. As we continue our journey at CSIR-IICB in collaboration with the Swasthaniketan, we remain optimistic to bring out a comprehensive palliative care and treatment for the cancer patients of the country based on the Indian variety.
We are interested in indigenous organisms like cyanobacteria, and it all started when one of my students on a trip to Lakshadweep, out of curiosity, isolated a culture. We have since been maintaining it in our lab, and we have named it as LK, stands for Lakshadweep, and we have sequenced it 3 times already, and we have done a lot of metagenomic and comparative genomic analysis to prove that this is a novel species, and we have named it as Leptolimbia IICBca after our institute's name, and the strain name is called as LK. And we have also studied the metabolites for its bioremediation and antibacterial and anti-cancerous properties, and it has lot of potential in this area. And this organism also shows tremendous bioremediation properties. When the culture extracts or the dead cells are treated with a strong acid, it converts it into alkaline media within no time.
At CSIR-IICB, we have developed a new paradigm in drug discovery. It's called PROTAC technology, or proteolysis degrading chimera, or in short degraders. Conventionally, we have small molecules and that's been targeted to inhibit certain proteins which has been implicated in disease condition. So, instead of inhibiting a protein target, so here what PROTAC does is PROTAC degrades that protein. So, how it degrades? It takes the help of our endogenous degrading system, which is called ubiquitin proteasomal system, and take the help of E3 ubiquitin ligase enzymes. So, PROTAC basically is a heterobifunctional molecules. It has two active ligands, one targeting a E3 ligase enzyme like cereblon or VHL, and another ligand which targets a unwanted protein which we want to degrade. Now, these two ligands are joined by a linker. Now, here the length of the linker and the geometry of the linker is essential because it brings the E3 ligase enzyme in close proximity to the unwanted protein which we want to degrade. At IICB, we have developed a PROTAC platform to degrade ASK1 protein. Now, the overexpression of ASK1 protein has been implicated in non-alcoholic fatty liver diseases, and it is a validated drug target for which many big pharma companies working. Now, what we did at IICB is we have developed a PROTAC which can selectively degrade ASK1 protein, and we have shown that it can be an effective and alternative strategy in the management of non-alcoholic fatty liver diseases. Now, these PROTAC platform can also be extended to other therapeutic platform, for example, in cancer, in case of antimicrobial diseases, because we know that in these platform the drugs are becoming resistant because of mutation in the protein. So, PROTAC can work to basically degrade these mutated proteins.
We are actively engaged in developing cost effective industry friendly process for the synthesis of KSM, key starting material, and its application for the synthesis of high valued generic drug molecules under the mission of Atmanirbhar Bharat. Apart from this translational research, we are also actively involved in basic and fundamental research in the area of electrocatalysis and organocatalysis. Very recently, we have developed a very cost effective process for the synthesis of 2,7-dihydroxyfluorinone, is a KISM for the synthesis of telurone, which has broad spectrum of antiviral activity, which act as a interferon inducer agents. Apart from this antiviral mission, we have also developed a cost effective industry friendly process for the synthesis of KSM like indole 3-carboxylic acid from easily available starting material in indigenous way. And what is this indole 3-carboxylic acid? What is the use of it? It can be used, and we have used for the synthesis of a very important drug molecule that is called topisetron, which is actually regularly prescribed by the doctor for the chemotherapeutic patient to avoid vomiting and nausea. On the other hand, we also have developed a process for the synthesis of palbociclib in the mission of making cancer care affordable. And this palbociclib, we have developed a process, very cost effective process, not only palbociclib but also its intermediate. It is which actually used for the treatment of HER2 negative and HER2 positive breast cancer for the women. It is a selective selective kinase inhibitor. Apart from this healthcare mission, we are also engaged in developing various cost effective process for the synthesis of agrochemicals. With that, thank you.